Thanks to the three of you for putting this fantastic seminar series together. I'm very excited to introduce our speakers today. Stephanie Powell, she's an associate professor and an associate dean for research at Florida State University. Stephanie is a global change ecologist who investigates the diversity and productivity of forests, savannas, and grasslands, and how they are impacted by climate change. With that introduction, I will turn it over to you, Stephanie. Welcome.
Thank you. Thank you so much for that introduction, Michael. I'm excited to be here and excited to share my work. So the title of my talk today is Understanding Ecosystem Functions Across Scales with Eyes in the Sky and Boots on the Ground. My slides aren't advanced. Okay, so to start off with, we are living, as we all know, in unprecedented times when anthropogenic climate change is causing a rapid rise in global surface temperatures. But the reality of this climate change is that it's not a single squiggly line. The pace and magnitude of climate change varies across space and time, so that different locations on the Earth are experiencing climate change at different rates. And this means that those species, communities, and ecosystem functions in those different locations are going to be impacted by climate change differently. So, for example, in this animation, you can see that high latitudes are warming more and much faster than low latitude tropical regions, which are thought to be generally more stable. So given this rapid global change, a general question that motivates my research is which species and communities will persist in the future with climate change, and how will this alter the habitats and ecosystems that sustain us? So more specifically, questions that my research addresses are how and why are plant species distributed along both temporal and spatial environmental gradients? And what are the consequences of this for ecosystem functions such as phenology and productivity? As Michael said, I consider myself a global change ecologist and a biogeographer. My approach generally combines site-specific field surveys in tropical forests, in savannas, in grassland systems, with geospatial technology. So for example, drone and satellite imaging, and statistical modeling, such as time series modeling or working with spatially autocorrelated data. My talk today is in two parts. So first I'm going to talk about tropical forests, and then the second part of my talk is about grasslands. These are, I acknowledge, two seemingly very different habitats and ecosystems, but both are biodiverse, they are highly productive, and they're both increasingly threatened habitats. So the first part of my talk, I'm going to talk about tropical forest phenology and productivity. I've decided to present a range of work that I've done over the years today, but I'll end by talking about a current NSF-funded project that I have. Two of the best examples of climate change impacts on species and communities are species range shifts, so shifts in their distributions and where they occur, and shifts in their phenology, the timing of life cycle events such as leaf unfolding, flowering, etc. And both of these should have really large impacts on human well-being, on ecosystem health, and on climate feedbacks between the biosphere and atmosphere. The general pattern that's emerging with species range shifts is what's called uphill and poleward. So with climate change, higher latitudes and higher elevations are starting to warm, and species are essentially tracking those warming temperatures, moving uphill and poleward. There's also stronger evidence at the leading edge, so more evidence. Can people see my mouse? So on the top right here, more evidence of species colonizing new regions that are now favorable for their existence. There's less evidence of species actually going extinct because habitats are no longer favorable. As opposed to physically moving, species can also stay in one place and they can alter their behavior or activity. there's mounting evidence for what's called spring advancement, and that's the earlier occurrence of spring events like plant bud bursts, flowering, and with birds and insects, for example, migration times. So on the bottom left here, this is the meta-analysis I was a part of, where we showed that on average, plants are advancing their flowering and leafing about 5 to 7 days earlier per degree Celsius of warming. But most of this evidence is from Northern Hemisphere temperate, boreal, and Arctic regions. So here, this is plotting in both the gray and the blue dots. These are locations of where many of our long-term phenological records are from. And it's plotted here against mean annual temperature and mean annual precipitation of each of these sites. So what you're seeing is, if you look at temperature right here, if you hopefully you can see my mouse, but this box right here is showing that most of our long-term phenology records occur within a really narrow range of temperatures, and to some degree, a somewhat narrow range of temperature of precipitation as well. In this example, there's only one tropical record. This is Luquillo in Puerto Rico. What that means is we have few long-term phenology records, at least relative to records in temperate regions and higher latitudes. And this can lead to a bias in our understanding of how climate change is affecting species in tropical regions. So for example, in the tropics, there's no comparable spring advance. These are habitats that are generally warm year-round. We have a poor understanding of tropical species' sensitivities to climate change. This is showing on the y-axis, I always say number, it's the percent of species that are significantly responding to increases in temperature in their first flower or first leaf date. This is plotted against the mean annual temperature of each of these locations. And so what you're seeing here is that these warmer sites right here, and this is Luquillo, again, that site in Puerto Rico, the tropical site, there are fewer species that are responding to temperature. Instead, tropical forests and tropical regions may be more sensitive to changes in light availability associated with changes in cloud cover. So this is showing a global perspective on factors that we think of as limiting to plant activity. So in the high latitudes, for example, these are cold regions that are thought to be most limited by temperature. Whereas if you look at the US Southwest arid regions, these are thought to be most limited by water. And then you can look at large areas of tropical forests in the Amazon and Congo Basin. Tropical regions are generally warm and wet year-round, and so they're thought to be most limited by radiation or light availability. There's a number of reasons why our understanding from temperate regions may be inadequate for understanding how phenology and productivity may respond to climate change in the tropics. So in the tropics, these ecosystem functions can vary as a function of differences among species. So tropical regions are one of the most species-rich habitats on Earth. That means not only are there numerous species in these different ecosystems, but many of them have different phenological and different growth strategies. Because of the number of different species and the different growth strategies, we might expect that they respond to multiple climatic factors and how those climatic factors interact. And lastly, the spatial and temporal scales that we measure climate and climate change can affect our understanding. Tropical forests, they cycle more carbon, water, and energy than any other biome on Earth. And like I said, they're among the most diverse habitats on our planet. So I'm going to show an example from just one tropical forest site in Panama. This is Barro Colorado Island in Panama, and I'm going to talk about this site for a little bit, the first part of this talk. Here, what you're seeing is a remarkable long-term multi-decadal increase in flowering activity at this site. And it's been increasing at a rate of about 3% each year, so producing more flowers. In addition to this long-term trend, you can see that it's also a really dynamic record. So there are these peaks that occur during certain years. And these two large peaks occurred during two strong El Nino events. At this site in Panama, El Ninos are associated with less cloud cover. It's associated with changes in the ITCZ, so less cloud cover. That means it's sunnier, it's drier, and it's warmer. And those conditions are associated with these huge peaks in flowering right here. These long-term tropical flower records, they're important for understanding phenology, like I just talked about. They can also tell us about the reproductive capacity of these forests and can indicate changes in productivity. So next, I'm going to show how different climatic factors can be important on different timescales. So on the left here, this is looking at seasonal timescales, how month-to-month variation in climate affects flower production. And what you're seeing is on a seasonal timescale, there's more flowers when it's warmer. So flowers increase with warmer air temperatures, and there's more flowers when there's fewer clouds. So essentially when it's sunnier, there are more flowers. Sorry, there are fewer. I said that there are fewer flowers when there are more clouds. I said the correct thing, but gestured at the wrong part of the graph. If we look at the interannual timescales, though, so this is year-to-year variation in climate and in flower production. We're seeing once again warmer temperatures, more flowers, but now rainfall is more important. So during wetter years, there are more flowers. A couple points I want to make here. One is that temperature is always in the best fit models on these different timescales. And the second thing is that the differences from seasonal to interannual timescales is not so much due to a species adjusting its physiology. So it's not a species becoming more sensitive to light on, month to month and more sensitive to water year to year. It's actually that we need to understand the magnitude and variability of how the climate itself is actually changing. So in contrast to conventional thinking, we think of tropical habitats as warm year-round and relatively stable. I've shown that tropical species are sensitive to temperature, but different combinations of temperature, rainfall, and cloud cover were important for predicting flower production across these different timescales. And again, when we're trying to understand the vulnerability of species to climate change, we need to understand not only their physiological tolerance, but how the climate itself is actually changing, the pace and magnitude of climate change. Most studies use air temperature to understand species distributions, phenology, and ecosystem functions. So I have an example here. This is from the Big Island of Hawaii. This is showing air temperature on the left, land surface temperature on the right. The difference that you're seeing, if we start with air temperature, you're seeing these nice smooth gradients. And that's because many air temperature data sets are interpolated, sometimes between pretty sparse meteorological stations. So what you're seeing is basically the lapse rate here. It's being interpolated between these meteorological stations, and you're seeing cooler temperatures and high elevations, warmer temperatures at low elevations. On the right, this is land surface temperature. This is the temperature that's taking into account the differences in surfaces that interact with air temperature differently. Over vegetated areas, so there's an area right here that I'll talk about. This is an area of tropical forest. This is canopy temperature, and this is the temperature that those trees are actually experiencing. And you can see not only is it a very different spatial pattern when you look at air temperature versus land surface temperature. I forgot to say on the right here, this land surface temperature, it's directly observed. So it's not interpolated. It's directly observed from a satellite called VOTIS or sensor. So this is not only a very different spatial pattern that you're seeing, but if you look at the legend here, also a very different magnitude of temperature differences. Plant physiologists have been aware of the difference between air temperature and organismal temperature for a long time. So leaf temperature, as opposed to air temperature, is known to be a fundamental control on rates of photosynthesis, respiration, and other processes. On the bottom here is an example of 4 tropical tree species and how their photosynthesis responds to differences in leaf temperature. But it's unclear if these leaf level measurements scale up to canopy level processes. So many of these leaf temperature measurements, they are short-term snapshots, and they also miss a lot of heterogeneity within a canopy. It's the canopy scale that should be most relevant for understanding processes such as primary productivity. And so that productivity basically represents the amount of carbon that's fixed through the process of photosynthesis. So looking at this canopy here, this is that same site I've been talking about, Barro Colorado Island on Panama. What I want you to imagine is that the sun comes up, it hits the top of the canopy, and that radiation warms those leaves up. very quickly above air temperature. Then there are traits associated with stomatal controls, leaf traits such as shape, angle, leaf size. Pubescence is a leaf surface trait that affect how much that leaf or tree can cool down. And there are also traits associated with the canopy. So canopy roughness, height, density of the canopy, and microclimate conditions such as wind and whether the canopy or the leaves are in full sun versus shade that affect canopy temperatures. So this is a nice green canopy that we see with our own eyes. This is a thermal camera in the foreground that you're seeing that we mounted. This here is what the thermal camera sees. This is the 8 to 14 micrometer wavelengths picking up thermal radiation from the canopy. And this is essentially showing that canopy temperatures are responding to heat and water exchange between the canopy and atmosphere. So with this thermal camera, we can take near continuous measurements of this canopy here in Panama. And canopy temperatures are very dynamic. So what I'm showing you here are 2 consecutive mornings, same time of day, it's 10 a.m., 2 different mornings. And you can see a very different spatial pattern across the canopy. So different hot spots are showing up. And once again, if you look at the legend, a different magnitude of temperature change. So here, this is showing how much canopy temperatures depart from air temperatures at this site. On average, canopy temperatures were about 2 degrees Celsius warmer, but could be as much as 7 to 8 degrees Celsius warmer than air temperature. So that's a huge difference. And that is the temperature that the tree itself is actually experiencing. So what you're seeing here is that canopy temperatures warm at about 40% faster than air temperatures. And at higher temperatures, the departure is even greater, and there's also more variability at higher temperatures. Here, what I'm plotting is gross primary productivity. So this is primary productivity, not accounting for what's lost through respiration, and how this primary productivity responds to canopy temperatures. And what you're seeing is at about 32 degrees Celsius, there's a decline in productivity where it gets too hot for the canopy to be as productive as it was at cooler temperatures. If we look at GPP against air temperature, you can see that air temperature does not capture this decline in productivity. Canopy temperatures were a better predictor of productivity than air temperature or vapor pressure deficit. And vapor pressure deficit is a measure of the aridity of the air. And so when the air gets too dry, plants will close their stomata and they're not as productive, essentially. But we're showing that canopy temperatures were a better predictor than air temperature or vapor pressure deficit. We can also look at canopy temperatures across different sites. So on the left here, this is a semi-arid temperate coniferous site, Metolius in Oregon. And on the bottom, this is showing the relative importance of these 4 different variables that are known to affect how much canopy temperatures depart from air temperatures. And so what you're seeing here is that at this semi-arid site, vapor pressure deficit is the most important variable that's predicting how much canopy temperatures are elevated above air temperatures. So again, that's how dry that air is. This is another temperate coniferous site, but it's a moist site. And at this site that's not as water limited, it's net radiation. That's the most important variable for predicting how much canopy temperatures are elevated above air temperatures. And then this is that same site I've been talking about, a tropical forest site in Panama, a moist tropical broadleaf forest site. And here, once again, it's radiation that's the most important variable. So what this suggests is that in the future, the relationship between canopy temperatures and productivity will change across sites. And the point that I want to make is that from an energy balance perspective, we can already quantify how much these different abiotic controls will affect leaf or canopy temperatures. What energy balance theory cannot tell us is the relative importance of these factors across different sites. And so once again, it's understanding the climate at each of these sites and how the climate is actually changing at these different sites. There's one more point that I wanted to make, and it's that thinking about the future from a global change perspective, these two sites that are most sensitive to radiation, there will only be long-term changes if there are also changes in cloud cover, because this is radiation from the sun. That's the most important variable for understanding canopy temperatures. So unless there are long-term changes in cloud cover, we would not expect long-term changes in these factors affecting canopy temperatures. And at this site, at Barro Colorado in Panama, I've shown that there is no, or there, from the data I looked at, there doesn't appear to be any long-term changes in cloud cover at this site. Vapor pressure deficit, on the other hand, is tightly linked to air temperature. So many of these arid sites that are really sensitive to the aridity of the air and really sensitive to vapor pressure deficits, those sites may be more vulnerable to future warming temperatures. So I've mainly talked about abiotic controls on canopy temperatures. I did, allude to different leaf traits like leaf size, for example, leaf angle affecting canopy temperatures. So do canopy temperatures also vary by species? This is a project on the Big Island of Hawaii. This is led by my current postdoc, Shannon Bayless. And this is a 4-hectare plot. It's a wet forest site on the windward side of the Big Island. And using high resolution drone images, so this is about 20 centimeter resolution drone images, she is mapped throughout the entire 4-hectare plot, every crown in the plot, and identified them to species. So here, this is an example of the two dominant species in this plot, Ohia on top and Koa on the bottom, which are the two dominant species. And you can see in these pictures, they have very different leaf sizes, leaf angles, different leaf traits, and also very different canopy structure as well. So once again, at this tropical forest site, this is showing down here, these are the relative effects of these different abiotic factors on how much canopy, in this case, crown temperatures, because we've mapped every single crown. So how much each crown temperature departs from air temperature, which we call T-diff. So it's net radiation, again, at this tropical site that's most important. But the thing that we're showing here is that species differences account for 18% of the variation in T-diff that we're seeing across the canopy. So there are some really important differences by species. If we look on the right here, these two, Ohia and Koa, are again the two dominant species. These are two other subdominants. But you can see if we look at individual crowns for these species, there might also be some differences in their sensitivity to the aridity of the air. This is vapor pressure deficit again. So Ohia might be slightly more sensitive to aridity versus Koa because of traits, leaf traits associated with this species. So the last thing that I want to show here is this figure right here. And if we just focus on the x-axis first, these are crown temperatures, again, of the two dominant species on the left. These are the two other subdominant species. And these are the same ambient temperatures that all of these species are experiencing. So this is all from the same time of day, about 10:50 a.m. in the morning, same habitat, same plot, same ambient temperatures. But if you look across the x-axis, they're all experiencing very different crown temperatures. So the temperature that they're actually experiencing can be very different from the air and also very different from each other. I'm plotting near infrared reflectance against crown temperature. Near infrared reflectance is a remote sensing proxy for photosynthesis and productivity. And this also suggests that these different species might have very different photosynthetic responses to crown temperatures as well. So this is all part of a larger NSF funded project that's about that's trying to understand how climate change will affect the phenology and productivity of two different tropical forest habitats. I've mainly focused on talking about this wet site, Laupahoehoe, and I mainly focused on talking about the thermal work that we're doing. But we also are taking the same measurements at a nearby dry forest site. And we have been measuring monthly leaf and reproductive litter fall for more than 10 years at the wet forest site at least. Woody growth, a full census is done of these sites every 5 years. And we also have our drone hyperspectral LiDAR and thermal images throughout 1 year. So next I want to move on to talking about grassland diversity and distribution. And this is an NSF Macrosystems funded project. Human civilization depends on grasses. So many of our staple crops, wheat, rice, corn, for example, these are all species in the grass family. Grassy biomes, which include grasslands and savannas, account for about 40% of the global land surface, about 30% of terrestrial productivity, and they're also, like I said, a staple for human sustenance. Grasses are also one of the largest flowering plant families, with about 11,000 different species just in this one family. Where and what are what I've called grassy biomes? So anywhere where the ground layer is dominated by species in the Poaceae family. In this mapping exercise I'm showing at the bottom, we used a definition of more than 50% cover. Anywhere where the ground layer was dominated by more than 50% cover of a grass species, we mapped as a grassy biome or grassy habitat. These are areas that have sparse to no tree cover. They include steppe grasslands and prairies, but also temperate woodlands and tropical savannas. So sparse to no tree cover, but in some cases there can be some trees. The key is that these trees are sparse enough that it's an open canopy highlight environment so that the understory is still dominated by grasses. The question of where these grassy biomes are is more complicated than one might think. So using classic biogeographic biome mapping based on climate falls short when we're trying to understand where grasslands and savannas are. This map right here is showing in all of these black regions. These are classified as ecosystem uncertain if you use just strictly climate definitions for where we think grasslands or woody habitats might be. So these are regions just based on climate, could be either grassland or woody dominated. To understand where grassy systems are, in addition to climate, we need to understand factors such as fire, herbivory, soils, and plant traits for understanding the distribution of the grassy biome. The misclassification of the grassy biome has consequences for conservation. And so these are two somewhat recent high-profile examples of papers that have argued and mapped where we should be planting more trees as a climate change mitigation strategy. So if we could just plant more trees in some of these areas, for example, I'm in North Florida right now, and this has been characterized as poorly stocked timberland. But in fact, much of this area in the southeast here is longleaf pine savanna. And so these are areas that if planted with trees and converted to a forest, it would come at a cost to the high biodiversity that's supported in many of these ecosystems. Grasses and grassy systems are often misclassified and understudied compared to trees and forests. And like I said, this comes at a cost to the high biodiversity and ecosystem function of the grassy biome that's supported by the high light open canopy environment. And grasses are also poorly represented in trait databases and in global land models. The most common trait that people use to classify grasses and grasslands is based on photosynthetic pathway, so C3 versus C4. C3 and C4 are basically two different ways of photosynthesizing, and each of these are advantageous in different environments. So this is sort of a classic biogeographic map of the distribution of different grasslands, with C4 grasses more common in the lower latitudes and warm environments, and C3 grasses occurring in high latitude, cooler environments. But C4 photosynthesis has evolved independently more than 20 different times in different evolutionary lineages. And what that means is that this trait wasn't simply passed down, but evolved in distinct ecological contexts. So C4 evolved in different species, mostly grasses, at different times, in different places, and under different ecological and environmental contexts. That means that a simple C3-C4 distinction will mask a lot of important grassland diversity and ecosystem functions. So here, this map is showing, instead of just C3-C4, the huge diversity of major grass lineages, and you can see how they sort along different environmental gradients. Of all of these major grass evolutionary lineages, 3 of them are globally dominant. So it's Andropogones, Chloroides, and the Puides that are globally dominant. And right here, this is mapping the distinct climate space that these 3 grass lineages occur in. Andropogones tend to occur in warm and wet regions that experience frequent fire. They're really dominant here where I am in North Florida. Chloroides tend to occur in warm and dry regions. So you'll see them more in the arid regions of the North America here. And the Puides tend to occur in cool and dry regions. I'm going to talk about an NSF-funded project we have that's focusing on the Great Plains region right here. And this is where we have a dominance of all 3 of these lineages. So in the U.S. Great Plains, how are lineages distributed along environmental gradients? What are ecological growth strategies, so fast versus slow growth strategies, of these evolutionary lineages in different environments? And are these different growth strategies associated with differences in ecosystem functions, such as rates of productivity? So this is a large collaborative project with many different PIs, but I wanted to highlight the graduate students who are working on this project. It's Choling Ho from Penn, Ryan Donnelly from Kansas State, and my graduate student, Ryan Slapikas, here at FSU. What our group has done is measured, and again, the grad students have done the heavy lifting here. We've measured just a ton of different functional traits for a number of different grass species across different sites in the Great Plains. We've measured physiological traits, leaf structural traits, biochemical traits, hydraulic traits, and we've also measured spectra. So that's the leaf reflectance from these different grass species and different lineages. And I'm going to talk about the spectra a little bit more than the traits, although I'll talk about the traits a little bit. The spectra are really promising for allowing us to scale across landscapes and to the extent that managers are often working at to understand ecosystem functions and how these grasslands may be responding to climate change. So we sampled 66 grass species from 5 ecologically distinct sites, measured a suite of different leaf traits and leaf reflectance at peak greenness. So our 2 most northern sites, North Dakota and Minnesota, both of these are relatively cooler sites. One is dry and one is mesic, more wet. Further south, our next 2 sites are Colorado and Kansas, a little bit warmer than North Dakota and Minnesota. And again, one is dry and one is more wet. And then our most southern site is in New Mexico. This is a desert grassland that is hot and dry. So I'll talk about the traits just a little bit, and then I'll move on to talking about the reflectance spectrum. So here what I'm showing is a principal components analysis. A PCA, what it does is basically it will reduce the many different traits that we measured, and it will reduce all of those sometimes correlated traits into 2 uncorrelated axes of variation, or at least I'm showing 2. So it reduces all of those traits into these 2 uncorrelated axes of variation here. And each of these points that you're seeing are different species that we've collected trait information for. The different shapes are the different sites, and the different colors are the different lineages. So for example, you will see more squares on this side of PC axis number 1. You'll see more triangles on this side of axis number 1. If you look at axis number 2, you'll see more red colors up here, so more of the Puidae lineage. And if you look at the bottom of, towards the bottom of this axis, you'll see more of the yellow color, the Andropogones. So PC1 is basically capturing differences across sites, and PC2 is basically capturing differences across lineages. I know there's a lot on this graph. There's about to be more. So we can also plot, these are what we call loadings on this PCA analysis. So we can also look at how these different trait values fall along these 2 axes that are capturing variation across sites and variation across lineages. So I'm just going to point out 3 traits as an example to understand some of the ecological strategies of these lineages. VC max is a trait that tells us about photosynthetic capacity. So the species that are on this side of this graph tend to have high photosynthetic capacity. So for example, the Puidae species tend to have high photosynthetic capacity. This trait right here is turgor lost point. It's a hydraulic trait. And species down here tend to be more drought tolerant. So for example, Anthropogones tend to be more drought tolerant. This trait right here is leaf dry matter content. It's a leaf structural trait, and it can tell us that species on this side of this plot tend to be more resource acquisitive, and they indicate more faster growth rates. And so the general pattern that we're seeing is that, for example, the poets here in the red, they tend to have high photosynthetic capacity, and they tend to be less drought tolerant. If we look at the Chlorodoides, so those are in the pink, the Chlorodoides tend to load onto lower photosynthetic capacity and higher drought tolerance. And so we can start to see some different ecological strategies emerge. And something I forgot to say is with these traits, I know there's a lot on this figure right here, it's important to look at how they co-vary. Because looking at traits in isolation doesn't account for how traits might be correlated with each other. So next I want to talk about the leaf reflectance. Leaf reflectance, like I said, is really promising for potentially being a scalable feature of ecosystems. Here's an example of just 2 different species. These aren't grasses, unfortunately, but just 2, hypothetical species for the sake of this illustration. This right here is indicating that they have different biochemistry. This is showing a cross-section of these leaves. So because of differences in the internal cell structure of these leaves, they may absorb, scatter, and then reflect light differently, as well as because of their different biochemistry, they may also reflect light differently. What you're seeing here is a classic leaf reflectance spectra. So this is the electromagnetic wavelength. These are from the visible all the way to the shortwave infrared of light. And this on the y-axis would be percent reflectance. And this is an evolutionary tree here, or a phylogenetic tree. And this is suggesting that species that are more closely related to each other, like species in this area, will have a more similar spectra. And species over here might have more similar spectra here to the yellow, for example. So this spectra that we can measure at the leaf level, it's the same spectra that we can measure for a plot. And it's the same spectra that we would measure from an airborne or satellite sensor, giving us information across landscapes. Again, at the extent that many land managers care about. So this is work led by my graduate student, Ryan Sapikis. And what he's shown using something called a linear discriminant analysis is that leaf spectra grouped by evolutionary lineage, these 3 lineage, with very, very high accuracy. So we had over 94% accuracy. This is just the representative reflectance spectra for these 3 lineages, Anthropogony, the Chlorodoides, and the poets here. Each of these regions of the electromagnetic spectrum is thought to be responsive to different leaf traits. So in the visible portion, this reflectance is most responsive to differences in leaf pigments like chlorophylls and carotenoids. In the near infrared region, reflectance here is most responsive to differences in cell structure, in addition to some leaf surface traits. And the shortwave infrared region is most sensitive to leaf biochemistry, like nitrogen content, for example, but also proteins, lignins, cellulose, and water content. And so he's shown that the near infrared region was the most important region for discriminating lineages based on leaf reflectance spectra. And so here's an example of how we measured the leaf reflectance spectra. This is a leaf clip that we have attached to our spectrometer. And there's a little bit of nuance in what were the most important regions, so I'm happy to answer questions about that later. Leaf spectra, driven by differences in these leaf traits, can also vary in different environments. So I'm showing here that they're very strongly conserved by evolutionary lineage, that if you just know their lineage, you have a good chance of identifying which evolutionary lineage that spectra is from. But spectra can also vary across different environments. to what degree are grass leaf spectra phylogenetically conserved or evolutionarily conserved versus environmentally determined? So despite large environmental differences across these different sites, evolutionary lineage explained more variation in leaf spectra than site differences. So about 62% compared to about 38%. So once again, these spectra are strongly predicted by if you just know the identity, the lineage identity of these different species. But if we limited our analyses to species that occur at more than one site actually explain more variation in spectra than lineage. And I should say that this is of all of the explained variants. So there is some unexplained variance in the spectra, but of all the explained variants, site explained about 72, so that's a pretty large proportion. 72% of variation in spectra, and lineage only explained about 28%. So what this suggests is that species with larger ranges, these are species that are occurring at more than one site, they're showing more plasticity in their spectra and their traits, perhaps maybe allowing them to be more generalists. The other thing that this suggests is that some of these traits are locally adapted or locally acclimated. So what a lot of people want to know is, can we scale up from leaf spectra to airborne or satellite imagery? Remote sensors have been doing something called spectral unmixing for decades. So here's an example of these three different surfaces that might show up in a remote sensing image. A would be bare ground, B would be water, and C would be vegetation. Oftentimes, the scale of a pixel that we get from a satellite or an airborne sensor is large enough where it contains a mixed spectral signature from different land surfaces, right? So this is a mixed pixel problem. When you want to know the proportional abundance of these different land surfaces, how do you do that when you have a mixed pixel? If you have a pure end member for each of these, meaning you know what the spectral reflectance is for something that just contains this land surface, then you can try to use linear algebra to decompose the fractional abundance of each of these in a mixed pixel. So here's another diagram that illustrates that. This down here might be a pixel containing these three different surfaces. Sunlight hits these surfaces, it reflects, the sensor captures that, measures that reflectance. And what you have here is a mixed pixel, but if you know the pure end members, what the reflectance of just that land surface looks like, you can unmix the fractional abundance. So remote sensors have been doing this for decades, but they've been doing it for things like bare ground versus vegetation versus water. And we are trying to do this with different grass evolutionary lineages. So it's quite a leap forward. If you look at this picture on the bottom right here, in a 1 meter square plot, for example, this looks like a pretty homogeneous, or if you just look at the entire picture of this grassy site, it looks like a pretty homogeneous green landscape. But that, what looks to us as a pretty homogeneous green landscape, actually contains a mixture of grasses from different evolutionary lineages. Can we try to identify those evolutionary lineages just based on their leaf reflectance? That's what we're trying to do. So scaling from leaf spectra to predict lineage composition in a plot here, range in accuracy from about 77% classification accuracy to about 40% depending on site. That's not quite good enough. Certainly 40% is not good enough. 77% is not quite good enough to create these large scale maps of these, to understand where these lineages are distributed and to understand their environmental preferences. But we're already improving this by, for example, using spectra from that site only, which ties back to the earlier results that I showed earlier. So instead of using all possible data from all sites, using only what we call end members or training data from a particular site that we're trying to classify and unmix. Maps are very cool, but mapping lineages and species in grasslands will help us understand how help us understand their environmental preferences and how they might shift with climate change. So once again, we're trying to scale from leaf reflectance to the plot scale all the way up to these large scale images across a site. So I'll just end by briefly showing this plot to talk about the potential to monitor and map invasive grasses. This is a grant proposal that we recently submitted just 2 weeks ago to the NSF Macro Systems program with Dan Park, who is at who is at Purdue. What I'm showing here is reflectance spectra from 4 1-meter square plots at a grassland site in Kansas. And you're seeing the spectra from invaded plots in the yellow and uninvaded plots in the green. So there seemed to be a difference in the spectra of plots with invasive species versus plots that have not been invaded. And going even further, plots that seem to have more invasive species. So this is a bromus species that's invasive in Canada. This is 30% cover, seems to have a different reflectance signature than plots that have less of that invasive species. So potentially huge promise in monitor and mapping invasive grass species. And we think that we might be able to do this because because traits of invasive species may be very different from native species in certain environments because they're trying to limit competitive exclusion. So the question is actually when and where. We don't expect this to always work, but in what types of environments, when and where, might spectra be very different between native and invasive species because of differences in those traits. And so I'll conclude with just 3 concluding thoughts here. We think of tropical forests as evergreen, very stable environments, but I've shown that they're actually highly dynamic, and that's in part due to species-specific sensitivities to even small changes in temperature. Grasslands are structurally simpler, but the taxonomic and evolutionary diversity within just one family has resulted in diverse ecological strategies. In both these systems, observations across scales from leaves to landscapes can provide new insight into how these ecosystems will be impacted by climate change. And so I'll end there. Lots of people to thank and acknowledge. This is a picture of my current graduate students. I didn't have a chance to talk about all the very cool work that they're doing, but they're all doing great stuff. My postdoc, Shannon Bayless, and my lab tech in Hawaii, Amberly Pagao. Thank you.
Wonderful. Thank you very much. Folks can go ahead and either raise their hand or unmute and ask a question. Oh, Ron?
Hi, thanks for an interesting talk. I might have missed it. Are you doing spectral analysis of, for instance, mixed grasslands like C3, C4 that might have temporal changes over the course of the season in the relative abundance and the spectra associated with that?
Yeah, that's a great question. So at a lot of these sites, there is they do change in certainly their activity. So their greenness and their reflectance will be impacted by that, and they also change in their relative abundance to some degree. We measured all of our traits and our spectra at peak greenness at every one of these sites. And so this peak greenness was based off of a model of long-term MODIS data, modeling their phenology and identifying this window of peak greenness. And we also coincided our measurements with when a neon airborne sensor was going to be flying so that we could have the associated images with when we were on the ground. So we feel like we control to some degree differences in phenology, for example. Your question about, they're changing abundances, that's something we're hoping if we can develop a really strong link between spectra and these species or lineages, we can then map those changing abundances. So they're not changing hugely, across the growing season or anything like that, but certainly across years, you might see differences. And that's something that if we can do this, will be a great way to monitor how that activity is changing.
Wonderful, thanks. David, would you like to ask your question?
Sure. Thanks, Stephanie. It was a great talk. Just a couple of questions about your mixture models and your process. With using the data from the NEON AOP Have you tried using N members directly from the imagery itself instead of the field collected?
No, we haven't. We've mainly stuck to the N members that we've collected, although we have experimented with different N members. So for example, I briefly alluded to using N members. I was trying not to say N members too much because it can
It's
be
okay.
a kind of technical term, but let's call it training data N members. If you use N members from a particular site, so you would think with something like this, throw as much data as possible at this, classification problem. But what we actually have shown, at least preliminary data has shown, is do not throw as much data as you have at it. Instead, if you limit your N members to just those collected at that site, we get better classification accuracy. The other thing that we've experimented with, and sorry, I'm losing track of the screen, but anyway, I was trying to talk to you, but the other thing we've experimented with is using leaf N members that we measured in the lab versus 1 meter square plot N members. And so the plot, and I think there's some interest in just knowing like how good can we do with just a single leaf? How well does that do? And that was the 77 to 40% classification accuracy. So it's like promising, but not great. When we use the plot N members that we measured in the field with our field spec, we were getting, I didn't want to show it because my grad student and I are still making sure that it's all solid, but we're getting higher than 90% accuracy, which I think is pretty cool, but I'm biased because it's my work. And so that's what we've been playing around with as far as N members. And it would make sense that the plot and plot spectra is going to be better because it's accounting for structural variation and leaf angle, et cetera, in the field.
Yeah, and you're getting soil background and things like that in there. Yeah, I'd like to talk to you more about that. But yeah, thank you very much.
Cool, great, yeah.
Ron, go ahead.
Since no one else is raising their hand, so are you correlating your observations with not just mean annual temperature and rainfall, but seasonality of precipitation, which should really affect the C3 versus C4 relative abundances?
So we haven't gotten to that point yet, but kind of just tie back to your first question. I think that is sort of the holy grail is if we can produce some reliable maps, and we need to get our classification accuracy like 80% or higher actually, then we can start to track those seasonal shifts in time and space, which I think will be really cool. But we haven't gotten there quite yet. But I absolutely agree. It's going to be the seasonality that's going to be really important to consider.
Lau, why don't you go ahead? I had a really general question, so I'll let you go, especially if you don't mind.
Yeah, of course. Thanks for your talk, Stephanie. My question is related about the grassland side of your talk and how. So I understand that your objective is to try to understand the grassland distributions, and the most mesic end of your grassland sites has mostly been converted to agriculture. How do you plan, or do you include these monospecific croplands into these analysis and extrapolations in space?
Yeah, that's a really good point. So I'll say for the purpose of our project and how we've defined it, we focused our study at these ecological sites. So they're LTER or they're NEON sites, and there is not, well, I shouldn't say that. At some of these sites, Kanza and Cedar Creek's one of our sites. There are different burn and grazing regimes. We've kept track of what the different treatments are at those different sites. But for the most part, these are well-managed ecological sites where we know what the grazing and burn treatments are. And that also does affect our classification accuracy a little bit too. But I think maybe to your larger point, what you're getting at is the reality is many of these grassy ecosystems, which I have an interest in, continuing to work in, are in fact managed, and they have people on them. They have, grazing, they have, different human uses on them. And so I think it'll be hugely important to try to understand that in addition to just the ecological questions.
All right. I know it's 4:30, but we also want to make sure that folks have a chance to ask Dr. Powell their questions. So John, why don't you go right ahead? Oh, John, you're muted. Sorry, can you unmute, please?
Yeah, sorry. Hey, Stephanie, I was really interested in the drone work that you were showing. That's sort of coming out of your lab from Hawaii. So all of that work is all top down when you say vapor pressure deficit on top of the OHIA versus the COA. Those metrics are based just from that top down view. So they don't get the whole canopy. They're just seeing what you're seeing from the drone, right? Is that basically what we're seeing on that?
Oh, yeah, is that yeah, so in general, yes, it is top down and you're seeing just top of the canopy. We actually have had some interesting results because it's actually so high resolution. So the resolution of the drone is going to, well, anyway, it is about 15 centimeter resolution. In some cases, we're actually getting a little bit into the canopy itself. And so that makes interpreting some of the data a little bit tricky. It's actually, from understanding the drivers of some of the things I've talked about, it'd almost be easier to limit ourselves to strictly sunlit leaves so we have a better understanding of the actual environment. But yeah, so I don't know if I answered your question, but
No,
for the most part,
you actually anticipated my second question. Was like, can I
Okay.
use those for sunlit leaves? And you're telling me no.
I know, I thought it was going to be that simple. So if you are somebody who uses drones and you're getting really high resolution, you're actually peeking into the canopy a bit, which complicates things. I mean, frankly, at higher resolution, it's a mixture, you know, so in some ways we're better able to subset our data. So what we've done is we've used an NDVI and near infrared filter. And this is what some people have done in other contexts as well. So if you choose an NDVI threshold and a near infrared threshold, you can be pretty confident that those are sunlit active leaves. So that's what we're doing.
There's not strawberry guava underneath those COA and
Oh, okay. So you know these sites, not at the study sites we're working at, but back to, you know, to Lau's point, actually, in reality, there's a ton of strawberry guava in those areas. And so, you know, if we really want this to be relevant to land managers, we do have to think about those things. So it would actually be very cool to see if invasive species have, you know, different spectral signatures, different thermal properties, et cetera. But no, not at our site.
Right, thanks.
Native dominated. In fact, yeah.
Paolo, would you like to go?
Great talk, Stephanie. Just a quick question. You did that analysis of canopy temperature and air temperature. Did you do it also for grasses or only for trees?
So far only in these forests.
Okay.
Yeah, yep.
Oh, very cool. Thank you.
Thanks.
And I think I'm next. Thank you. I really enjoyed your talk. Especially I enjoyed seeing the very different case studies you presented, like tropical and grasses and agriculture. And so I was wondering what your vision is for like the next set of projects. Like, do you envision doing some also in the tropics, or we have a lot of faculty working in agriculture, so I wanted to kind of give you a chance to speak a little bit about that if you want to. Thank
Yeah, I
you.
appreciate that question. So I am very interested in working in the grasslands of California. So California is my home state, and I think that there is, you know, huge potential to work with local managers, state, federal agencies, et cetera. And so I'm very interested in that, in continuing some of the remote sensing work in the grasslands there. I think it's great also for students to work locally, and there's just a ton of sites, you know, within a day's drive of Berkeley to work at. That being said, I hope it doesn't sound like I'm interested in too many things, but I certainly still love the trees for all I said about the tyranny of the trees. Hawaii is somewhere I've worked for a long time now. I don't even want to count because it makes me feel old. And so I think I'll continue long-term research in Hawaii, just a place that I have a connection to, but I also have a huge interest in working in Berkeley forests as well. Those are other nearby sites where I think it would be great to work with students. But I think to answer your question, I think hitting the ground running, I'd love to find some really cool grassland sites in California and continue some of the work in Hawaii long term.
Thank you. And Adrienne had to leave, so I'll just ask Lynn to ask a question.
Lynn, you might be muted.
Probably a good thing. I enjoyed your talk. And in the grasslands that you've been studying, you mentioned that composition is not too labile. It doesn't change that much quickly. And in the California grasslands, there are vast changes every year. So is that a benefit to what you want to do you think, or will it be a problem for you to overcome?
Oh, I think
Or I mean, is it an opportunity or a problem?
Yeah. That's a great way. I think it's an opportunity because I think in some ways what I was saying before is if we can develop these really strong links between the spectra and different species identities, then it doesn't matter if they change quickly. We just have to be there, you know, with a sensor to, I mean, it actually does depend on what kind of remote sensing imagery is available. But if you can develop that link, you can map that those changing distributions. So I think it's an opportunity that might slightly be limited with the technology at this point, but that's, you know, rapidly advancing.
Well, I wonder if you could do it by some subset of functional groups, you know, because the things people are interested in tend to run along broadleaf, excuse me, rare species, perennial grasses, and non-native annuals. So I'm wondering if that is a possibility.
Yeah, I think you're exactly right. Approaching it by looking at functional groups like perennial versus annual grasses. And I think once again, there's an opportunity because that should be some, you know, unique dynamics that we can pick up with the imagery.
And I had one last question. And I'm sorry that I'm not very knowledgeable about remote sensing, but it seems that a big thing for us, given both fire and biodiversity concerns in California, is how much biomass is actually on the ground, particularly as we get towards the fall, and how it's distributed in grassland. Turns out to be pretty important and pretty malleable with management tools. So I think having both those things would be great. Is that connected to what you're doing?
Yes, I'm glad you said that because I didn't talk much about, I kept, you know, implying that traits and spectra are related. We're actually looking at what exactly those relationships are for a bunch of different traits. Well, it turns out it depends on the trait, right? Biomass is one of those traits that we can predict pretty well with remote sensing data. So I think that there's another opportunity right there is to map and monitor changing biomass. Other traits like photosynthetic, or I don't know, like turgor loss point, that's a little bit trickier.
Thank you.
Thanks.
Well, if there are no more questions, I would like everyone to thank Dr. Pao. This is a great seminar, and I encourage everyone to join us for the seminar series. Next week, we have Josh Garcia.
We have one tomorrow, actually. Tomorrow is the next one. Do not forget, tomorrow at 2 o'clock.
There you go. Thank you. Thank you so much for everyone. And thank you, Dr. Pao.
Thank you so much for the opportunity to present my work.